Hi everyone. In this video, I'm going to teach you how to grow microbes on an agar plate like a microbiologist. So before we get to doing any of the experiments, we have to wash our hands. I want everyone to wash their hands to work on a clean surface. So wipe down your surface. That can be with Clorox wipes, Lysol. Just make sure that the surface you're working on is clean and your hands are clean. That's the most important thing, is that you wash your hands. What we're going to do today is to grow bacteria on an agar plate. So in your lab kit that you got from campus, if you open it, there's a lot of things, but I want everyone to take out the Ziploc that says streak plate technique. We are not doing streak plate technique. We are growing bacteria on a plate to do streak plate technique later. But this is the Ziploc I want everyone to take out. Again, it's called streak plate technique. And in that Ziploc, you will find 2 agar plates. I only want you to take out one of the agar plates. You'll use the other one later. So everyone take out one of the agar plates. And inside of that bag, inside of this bag, there's also a little tube. This is called a microcentrifuge tube. And this just has sterile water in it. So I'm going to teach you how to streak bacteria on an agar plate. This is called an agar plate. So if you can open it, try not to so that you don't contaminate it, but I just want everyone to see what it is. Agar is like a nutritious form of jello that we get microbes to grow on. So you can see that there's a lid, there's the agar plate, and we always, before doing any experiment or putting anything on an agar plate, you always want to label it. We always write on the agar side, not the lid, because you could potentially lose the lid. So that's what we're going to label. What you're going to do is you always label your plate on the sides, not the middle, because in the middle we want to see the microbial growth. So everyone should have a Sharpie in your lab kit, but if for any reason you don't, just use any marker. A Sharpie is preferred. And the way I label my agar plates is I always write my name. So I'm going to write Samar. And then you're going to write the date. So I'm writing the date 8/31, and then comma. I write whatever I'm planning to grow on here. So if you can imagine, if this was in a research lab, I would write the name of the bacteria I'm putting on here. You, for this lab, are going to decide what you want to grow on this plate. I'm going to talk about the things that you could grow on it, but for me, I decided to streak bacteria off of my phone and see what potentially is on there. So I'm just going to write phone on here. So the 3 things you want to label your plate with is your name, the date, and what you want to streak. So I put Smart 8/31 phone. You're going to choose something that you want to grow on the agar plate. The only rule is it can't be any body fluids. We're trying to keep you safe at home when you do these experiments. So you're not allowed to streak your nose, mucus, saliva, any bodily fluids. What you are allowed to streak is anything in the home that you're naturally exposed to, like your phones, pens, pencils, refrigerator, microwave. If you have a little pond water in the backyard, anything in nature, a food item, anything like that, you're allowed to streak on here. So I'm going to do my phone. Just the rule is no bodily fluids. Okay, the way we streak our plates is whenever I put my plate down, I always like to put my plate down on the table, agar side up. So I'm going to put this on the table. And in your lab kit, you have a bag that has a lot of things that look like sticks. There are blue loops, and then there are also these are sterile cotton swabs. So look for the sterile cotton swabs. Everyone should have them. You're going to take out a sterile cotton swab, and before opening it, it's a little bit transparent, so you can see the cotton side and the stick side. It's basically a sterile Q-tip. You want to open it at the stick side so that you don't contaminate the cotton, because the cotton side is where we're going to streak to pick up our microbes. So I'm going to open it at the stick side and grab it with my clean hands that I washed. And you're going to take your Q-tip and your sterile water. What you're going to do is you're going to open it and you're just going to dip your cotton in the water. I'm just getting it wet so that I can easily grab potential microbes off. Now, if you're streaking like a swimming pool or a pond, you don't need to wet it because that's naturally a wet surface. So there's my cotton swab that's wet, and what I'm going to do is I'm going to take my phone and I'm going to just streak all of it like this, just pick up all the microbes. I hope there's not a lot of microbes, but you're just trying to pick them up. And now I have my cotton swab. So you could have done this with anything at home. And so now that I have my potential bacteria on here, I want to get it on my plate. So you're going to take your hand and you're going to grab the agar side and you're just going to gently streak it everywhere. There's no rule right now. We're not doing streak plate technique. I'm going to talk about what that is later. I'm just basically taking your cotton and trying to get all those microbes on here. Now, a common mistake students do, just because you're not used to this, is they poke it. If you do poke it, it's okay, you're not supposed to, but just take out your stick and continue streaking all around. It basically feels like Jello here. So I streaked it out. I'm gonna cover my plate now. And so I'm gonna take my contaminated stick and I'm going to put it back in here. And I advise that for the remainder of the semester, anytime you contaminate things, you use them, put them in a Ziploc and then throw that Ziploc in the trash. So now I have my microbes on my agar plate, and now I have to incubate the agar plate. Incubate is basically just put it somewhere for the microbes to grow. Typically in hospital labs, we incubate plates at body temperature because we're trying to grow pathogens. Here, what you're going to do at home is you're going to incubate your plate at room temperature. What that means is you're going to look around your house and you're going to identify a location where you can let your plate sit or incubate for about 3 days. You all are choosing different things to streak, but it should take about 3 days to see microbial growth. Again, it's so different based on what you streak, but I would envision between 2 days to a week to see microbes growing on here. So for example, I'm gonna take my agar plate and I'm gonna set it on this table right here. This is the location I've chosen. It's high. You wanna choose a location that's room temperature, out of direct sunlight. You don't want direct sunlight hitting your plate, away from pets, away from children, and for your plate to stay untouched. What I want you to do is every day, so tomorrow morning, take a picture of your agar plate without opening it. Don't open it so you can pick it up. Just for your safety, I recommend that you not open it. Here's your plate. Take a picture and remember the incubation should always be agar side up. So again, this is what agar side up means. It means that the agar is up. We never incubate plates like this, always with the agar side up. I'm gonna leave it here for a few days and every day I'm gonna look at it until we see microbes. Once we have microbes on your plate, we're gonna use those microbes to do streak plate technique and a lot of other fun experiments. But first we have to grow microbes on those plates.